Have you ever looked at a sugar packet and just thought, it's sugar? Simple, right? Well, it turns out there's a whole secret language hidden inside its 3D shape. See, molecules with the exact same chemical formula can actually be completely different. And that little twist has massive consequences for, well, all of life. So today, we're going to decode that language.
So, the big question is, why should we even care if a few atoms are flipped around? I mean, really, what's the big deal about a simple flip? As you're about to see, it's a very, very big deal.
And here's the answer. Nature is incredibly picky. Our bodies run on this amazing molecular machinery, things like enzymes, and they have incredibly specific shapes. Just look at this. An enzyme might be a perfect fit for D-glucose, accepting it with open arms, but it will completely reject its mirror image, L-glucose. It's like trying to fit a left-handed glove on your right hand. It just doesn't work. This molecular precision is absolutely fundamental to all of biochemistry.
Okay, so let's start right there. We're going to dig into why this molecular shape isn't just a fun fact, it's profoundly important for life as we know it. Now that we have a feel for why this all matters, let's uncover the secret behind it all. We're going to look at the origin of all this amazing diversity, how one very special kind of carbon atom can create a whole universe of possibilities from the same basic ingredients. And here it is, the hero of our story, the origin of all this complexity. The chiral carbon. Now, a chiral carbon is simply a carbon atom that's bonded to 4 totally different groups. It's this asymmetry, this handedness, that's the key to unlocking all the incredible structural variety we see in sugars. And you know what? Almost every single monosaccharide has at least one of these. And check this out. This explosion of diversity is governed by a beautifully simple but powerful formula. 2 to the power of n. Where n is just the number of those chiral centers. Glucose has 4 of them. So if you do the math, 2 to the power of 4, you get a staggering 16 possible stereoisomers. That's 16 different molecules, all with the exact same chemical formula. Amazing. So, these isomers can be sorted into different families. The first big division we can make is between enantiomers. The best way to think about them is like your left and right hands. They're perfect mirror images of each other, but you just can't stack them perfectly on top of one another. In chemistry, we use a simple D and L system to tell them apart. A sugar gets the D label if its key chiral center matches D-glyceraldehyde, and L if it matches its mirror image. So let's apply that rule to our main molecule, glucose. We look at the chiral carbon that's farthest from the top of the chain, which in this case is carbon number 5. See that hydroxyl group, the OH? Since it's pointing to the right, this is D-glucose, the form that is overwhelmingly dominant in all living things. Its perfect mirror image, with the OH on the left, is L-glucose. Okay, next up we have the epimers. If enantiomers are mirror image twins, epimers are like close cousins. They aren't mirror images. Instead, they differ in the arrangement around just one single chiral carbon. For example, D-galactose is the C4 epimer of glucose. Only the 4th carbon is flipped. D-mannose is the C2 epimer. These are tiny changes, but they create molecules with completely distinct biological roles. But wait, the story doesn't end with these nice, neat, straight chains. In fact, that's not really how sugars exist in our bodies most of the time. So what happens when you put them in water, like in our cells? Things get even more interesting. What happens in a solution is that the linear chain actually folds back on itself. A hydroxyl group from way down the chain reaches up and attacks the carbonyl group at the top. This internal reaction literally snaps the molecule shut, forming a much more stable ring structure. This new ring is called a hemiacetal, and it's the form that sugars really prefer to be in. And this is where it gets really, really cool. The process of forming a ring, it creates a brand new chiral center out of thin air. The carbon that was originally part of that flat carbonyl group, that's carbon number 1 in glucose, is now bonded to 4 different groups. This special new center gets its own name. It's called the anomeric carbon. Because of this new chiral center, glucose now has 2 new faces, and we call these anomers. We label them alpha and beta. The rule is actually pretty simple. If that new hydroxyl group on the anomeric carbon points to the opposite side of the ring as the C6 group, it's alpha. If it's on the same side, you've got beta. Easy as that. But these ring forms aren't locked in place. They're not static. In a solution, the ring can actually pop open back into its straight chain form and then snap shut again. This means that alpha and beta glucose are constantly flipping back and forth in this beautiful dynamic equilibrium. The whole process is called mutation.
So with all these different forms floating around, linear chains, rings, alpha and beta versions, how do scientists keep track of it all? Let's take a quick look at the visual tools they use to draw and understand these incredibly complex shapes. First up, we have the Fischer projection. You can think of this as the essential 2D blueprint of the molecule. It flattens the whole thing out, which makes it really easy to see the configuration of each chiral center in that linear chain. The horizontal lines are imagined as popping out of the screen towards you, and the vertical lines are going away from you. Next, to help us visualize the ring structure, we use what's called the Haworth perspective. This is like a simple 3D sketch. It shows the ring as if it's flat, and you're viewing it from the side. And there's a handy rule of thumb here. Any group that was on the right side in the Fischer projection points down in the Haworth. But in reality, the ring isn't flat at all. To minimize strain and stop all its atoms from bumping into each other, it puckers up into a shape called the chair form. Now, this is our most accurate depiction of what glucose really looks like. It's the most stable form because it lets all the bulkiest groups stick out to the sides in what we call equatorial positions where they have plenty of room. So let's bring it all home. The crucial point here is that the simple formula C6H12O6 is just the very beginning of the story. From that one formula, we get our central molecule, D-glucose. And from there, we can define its mirror image enantiomer, its close cousin epimer, and its two different ring form anomers. It's really a case of one formula, but many, many different identities. And that really brings us to our final thought. The story of glucose shows us, pretty dramatically, how a tiny change in 3D shape can create a completely different biological reality. It just leaves you with this fascinating question. If a tiny flip can change everything for a simple sugar, what other incredible molecular secrets are hiding all around us, just waiting to be discovered, right in plain sight?